At some point, almost everyone is introduced to fossils. It might be in a classroom, a museum, or a natural area, or a scientific park. I have even had beach walkers join one of my classes in search for fossils, just for fun. And they collected more than my students. Whatever the circumstances, when an introduction to fossils happens, many people get excited. One of the interesting things about collecting fossils in western Minnesota is that one of the most common fossils are shark's teeth. Here's a Cretaceous shark's tooth, 70 million years old. They are abundant. In many cases, they are near the land surface. They are easily recognized, and they are easy to get excited about. Shark's teeth are totally cool. But shark's teeth in western Minnesota? We're in the geographic center of the continent, in the equidistant between the Atlantic and Pacific Oceans, and yet abundant shark's teeth. The answer, of course, is in geologic history. During the Cretaceous, 70 million years ago, while dinosaurs were roaming both to the east and west, a large epicontinental sea, which is an ocean on a continent, formed to the east of the Rockies. It was called the Western Interior Seaway. The animals that lived in the seaway were amazing. Of course, everything was big. Giant turtles, swimming reptiles, cephalopods, relatives of squid, and sharks of all sizes and ecological niches. And all of these animals left behind fairly well-preserved fossils. The sharks' eating habits were particularly well known because of the different types of teeth that you find. Some hunted fish and have pointed, straight, or curved teeth, but others would break open shellfish, and they had teeth that were peg-like. So fossils sound great. What could be the issue? The issue is this. If you are at an outcrop trying to teach students about Cretaceous geology, say sedimentology or stratigraphy or geologic history, even the briefest mention that shark's teeth have been found in that location, and the students are done listening. Done. The minute you mention shark teeth, they are looking for fossils and nothing else. Lecture over. But what the heck? I like shark teeth. I might as well look too. It's a problem I guess I have adapted to. The study of fossils, known as paleontology, is one of the most exciting components of field geology. In addition to being something that everyone can enjoy, paleontology also has a very practical side. Economic applications of paleontology began in the late 1700s when our friend, William Strata Smith, used fossils to find sedimentary layers that were useful for building stone. Today, fossils are used for finding petroleum-producing units. Recognizing and identifying fossils remains an important skill in resource development. But what exactly counts as a fossil? The word fossil comes from a Latin word, fossilis, meaning dug up. But a fossil is defined as the remains or traces of ancient life that are preserved in sediment or rock. That definition is interesting for a couple of reasons. First of all, a fossil does not have to be part or piece of an organism. Footprints are classified as fossils. That is, they are traces of ancient life. Another part of the definition to notice is that word preserved. What we've talked about in several lessons, from rivers to beaches and even for glaciers, is that sediment is always forming, even today. And whenever sediment is formed, organisms are also being buried. So when are they considered fossils? The answer is kind of tricky. For something to be a fossil, it has to be somewhat older than modern times. How old is that? Some sources suggest 10,000 years as a minimum age for a fossil, and refer to any partially fossilized specimen as a subfossil. For example, most people consider saber-toothed cat remains in the U.S. to be fossils, and they can be as young as 10,000 years old. But partially fossilized specimens can be especially valuable, since any organic still present can be a source of DNA samples or proteins or other biomolecules. As a rule of thumb, I tell students that if it does not smell, it's a fossil. If it smells, it's just dead stuff. Welcome to the world of paleontology. Fossils remain one of those things that continually attracts new people and perspectives to the geological sciences. As new people enter the field of paleontology, they bring both new ideas and new technologies to use in the analysis of fossils. Computer-based data files, CAT scans, different forms of modeling and animations, and of course, more detailed techniques to observe preserved body parts. But for newcomers, paleontology is a very tangible way to see the past, the organisms that lived in deep time, the environments that they lived in, the great difference between those environments and the environment of today. There's also an appreciation of the complex geologic processes that resulted in fossils being where you find them. When you find a fossil, you are the first person to uncover this piece of geologic history, and you join a long list of people who found joy in learning paleontology. You might also have found a clue that might lead to an important scientific or economic discovery. It's good to be aware of how rarely things get fossilized. That's not the impression that you might get from the abundance of fossils in museums or in the media. Only 250,000 species of fossils have been identified from all of Earth's history, whereas at least 3 to 4 times as many living species are known from this particular instant of geologic time on present-day Earth. One estimate suggests that less than 10% of all of the organisms that are alive today will be preserved as fossils. However, any single statistic is a little bit misleading because it combines some very fossil-friendly environments with other environments where fossils are very unlikely. In addition, animals with hard body parts have been much more likely to leave fossils than soft-bodied organisms, such as insects. So the fossil record, which is our complete set of fossils so far, is much more detailed for organisms with hard body parts that lived in environments favorable to preservation. Suffice it to say, there are a lot of organisms that have lived on this planet that have left no record at all. At least we haven't found them yet. So what conditions do favor fossilization? First and foremost is rapid burial or encasement. When an organism dies, it becomes fair game for scavengers. You observe this process every day. You know something that is lying by the side of the road attracts all types of scavengers. Scavengers that fly, scavengers that walk, scavengers that crawl. There are all types of scavengers out there, and they really lower the chances of preservation as a fossil. So you need an environment where the organism will be covered with sediment, or encased in tar, or frozen in ice. Even a lava flow can preserve evidence of an organism if the magma is cooling and encases the organism quickly. What is the common condition for fossilization in these cases? The absence of oxygen. Oxygen enables chemical reactions that will result in a breakdown of tissue, and oxygen will allow bacteria to grow. The result will be the destruction of the organism's remains. The final condition that encourages preservation of fossils is the presence of hard parts. The organism has bones, shells, or some other kind of rigid structure, it tends to be preserved. Soft tissue is very rarely preserved, although when it is, the fossils are just amazing. A lot of paleontologists spend a very large amount of time looking at teeth and shells. These are spinosaur teeth. The teeth of an animal would break off while it was hunting fish. These are the types of hard parts that frequently get buried and are preserved. The other thing that people quickly come to realize about fossils is that there are different ways fossils are preserved. The most impressive but least common method of fossilization is called complete preservation. This is the preservation of both soft tissue and hard parts. Preservation in amber received worldwide attention after the release of the book and movie Jurassic Park. It's a science fiction story based on the capture of genetic material in amber. Amber is fossilized tree resin. When a tree is distressed by fire, insects, disease, or other damage, it produces a resin in an attempt to heal the distress. Healthy trees don't produce this resin. Only distressed trees do. The resin dribbles along the trunk of the tree, and organisms are trapped in it. Here is an ant trapped in amber that was mined in the Dominican Republic. This makes amber preservation more amazing because you have fossils preserved only in resin on the surface of the bark. The resin has to be buried quickly to be preserved as amber. If amber resin is exposed to the atmosphere, it quickly turns to powder. Over time, the buried resin is transformed to a polymer. So if you have amber jewelry, you're not really wearing tree sap, you're wearing something that's chemically stable and relatively hard, and it won't turn to powder. This transformation process from resin to polymer occurs over substantial time. And if the amber isn't uncovered by erosion, or destroyed by weathering, or deformed by metamorphism, those small pieces may wind up in the hands of miners. You can find amber in a lot of places, but the most famous amber fossils come from 3 locations: the Dominican Republic, in sediment deposited 16 to 21 million years ago, the Baltic Sea region, in sediment deposited 40 to 60 million years ago, and Myanmar, frequently called Burmese amber, which formed 100 million years ago. Generally, at all 3 places, there was an abundance of trees in a nearshore environment, and fallen logs would be buried in shallow water or lagoons. The resin that encases organisms frequently flows over them relatively gently. Once encased, the organism is protected from decay because oxygen can't enter the resin. The result is spectacular preservation of ants, flies, occasionally lizards and frogs, and in the case of the Burmese amber, parts of dinosaurs are preserved. Ice is another way that complete preservation also occurs. One route to ice preservation is that the organism can be trapped in permafrost, or perennially frozen ground. Permafrost forms under cold climate conditions when soil freezes to depths, sometimes hundreds of feet deep, that are too deep to be thawed during the summer. But portions of the permafrost can be thawed by rapidly flowing water. Animals make their way to the water source and can be mired in adjacent wetlands. If the river changes course, both the entombing wetland sediment and the remains of the organism freeze. If they continue to remain permanently frozen, then the organism is preserved indefinitely. A second path to ice preservation is for organisms that expire in a harsh climate of a perennial snowfield or glacier. If snow rapidly covers the organism and over time turns to ice, the organism can be preserved completely, including things like stomach contents. There are a bunch of very famous fossils that were completely preserved thanks to freezing. The best preserved mammoth, named Lyuba, was frozen as a baby in Russian permafrost until its icy seal started to melt, and it was excavated in 2007. Ötzi, the famous Iceman, died on a snowfield on an alpine pass in northern Italy, not far from the Austrian border. Snow fell and he was buried for 6,000 years. His mummified remains were found by hikers in 1991. There are lots more that have been found and continue to be found. Up until the 1920s, there were groups of Arctic explorers that would excavate animals preserved in the Siberian permafrost using dog sleds. Because they had no refrigeration at the time, they would have to do the excavation in fairly cold conditions. The level of preservation was so good that they would feed the entrails of the mammoths to their dogs. 14,000-year-old dog food. Again, because the level of preservation they would have mammoth roasts for their rich donors. People actually ate the meat of the 14,000-year-old animal. Frequently what happens is, although the organism is completely preserved, it gets freeze-dried, a lot like the stuff in your freezer gets freezer-burned or freezer-dried. Ötzi the Iceman is sometimes considered to be a mummy, but his state of preservation was a result of natural freeze-drying. What happens during freeze-drying is that the mass of the tissue is greatly reduced and made leathery as water is removed from the hide. The hide can be more than 65% water. The proteins are compacted and become less pliable. While doing fieldwork in Antarctica, we found a completely preserved elephant seal. It wasn't very old, maybe only a couple of thousand years old, but the hide was impressively tough because of the freeze drying. Ötzi is a good example of complete preservation's importance in human history as well as geology. His accompanying artifacts provide insight into technology, culture, and religion of the Italian Alps 6,000 years ago. A third process I'll discuss that results in complete preservation is commonly referred to as pickling. This is where an organism dies in a chemical-rich environment that is oxygen-free and results in either encasement or burial. In food preparation, a brine is water with up to 20% salt in the solution, which is a common environment for pickling. Ordinary seawater has a salinity of 3.5%, but the Dead Sea has a salinity almost 10 times as great. Even so-called dead zones in ordinary ocean water, zones near the equator where water gets warm enough to retain less oxygen, can become locations for pickling of fossils. The lack of oxygen not only prevents decay, but also tends to protect against scavengers. Scavengers will avoid low oxygen waters. Over time, the chemicals in the brine that surrounds the organism replace the chemicals in the body tissues, resulting in preservation. This frequently occurs on the sea floor, where over time sediment will cover the fossil. Where does this happen? In saltwater brines, but also in oil seeps and in swamps rich in tannic acids. The process works exactly like pickling. You replace the natural fluids in a plant or animal with very stable chemicals, like salt water or a weak acid, like vinegar. When I moved to Minnesota, the neighbor brought over this jar of lutefisk, pickled cod. When I asked for an explanation, they said that the fish was preserved in lye, which is sodium hydroxide. That's scary, right? But the process works. The tissues of the cod are infused with a chemically stable lye. And with the absence of oxygen, there's no decay. In archaeology, which is the paleontology of our human past, pickling as a means of preservation comes up with the bogged people of Northern Europe. In some cases, it seems that young men dating back to 4,000 years ago were sacrificed to an earth mother, after which the bodies were tossed into peat bogs. In this case, the weak acids generated by the peat bogs provided the pickling agent. Not only did the tannic acid replace the fluids in the bodies, but it also dissolved the bone tissue in the preservation process. Occasionally, peat workers today will come across one of these preserved bodies. These bodies are frequently stained the teak color of the tannic acids and have the consistency of a gelatin. In some bodies, a chemical reaction between the hair and the acids will result in bright red colored hair on the mummy. Yes, it's a gruesome process, but in the geologic record, there are some absolutely amazing fossils preserved this way. One example is Tollenmann, found in Denmark in 1950. He died over 2,000 years ago. Initially, a doctor suggested he was murdered and cited several lines of evidence: a rope around the neck, a cut throat, a broken neck, and a skull fracture, all readily observed on the body. Over time, however, people are rethinking the cause of death because some of the injuries could have been burial damage. A much more common group of fossilization processes are those that only preserve the hard parts of an organism. Under most conditions, soft tissue decays over time, while the hard parts, shells, teeth, and bones, are preserved. Preservation can be through isolation or chemical alteration. Simply burying an organism in sediment will preserve it to some degree. Depending on the geologic processes involved, conditions can occur where hard parts are preserved. Marine sediments, of course, come to mind. Shells of animals, some fish bones and fish teeth are preserved with the original shell still intact. Here is a 3-million-year-old shell from Florida. On the Maryland shore of the Chesapeake Bay is a fossil location referred to as Scientist Cliffs or Calvert Cliffs, where there is abundance of different types of shells and shark's teeth that were buried perhaps 15 million years ago. Preservation of hard parts also occurs through petrification. The original meaning of petrify is to turn something to stone. A lot of why we have fossils involves the turning of organic tissue, or part of the organic tissue, into stone. I'll give you 3 ways organic tissue can be turned to stone. The first is permineralization, which occurs frequently with dinosaur bones. This is the deposition of mineral material in the pores of hard tissues. For example, once a hard part is buried, groundwater will move through it. Of course, the groundwater will have mineral material in it as it flows through the pores of the shells or bones of a buried organism. The through-flowing groundwater will deposit some of that mineral material in the pores of those hard parts. The minerals that are deposited can be silica or calcite or even iron oxide. What's key in permineralization is that the original tissue is still preserved, but it's made more resilient and harder than the original material. Here's a hadrosaur bone with permineralization. The reason this process occurs so frequently with dinosaur bones is that some dinosaur bones are very porous, much more porous than mammal bones. Here's a hadrosaur bone without permineralization. Dinosaur bones preserved in museums may look very dense, but theropod dinosaurs evolved with the same lightweight bone structure as that of birds. One of the lines of evidence of the relationship between birds and dinosaurs. The addition of silica to the structure through the permineralization helps preserve these bones, which otherwise would be likely to decay quickly, and actually makes the bones we find today more dense. My favorite type of permineralization is when pyrite's involved. The pyrite crystals have grown in the pores of the shells of these brachiopods. As the shell breaks apart, pyrite is left behind in the perfect shape of the shell. It's just amazing. A second type of petrification is total replacement. Replacement is the molecule by molecule exchange of the organic tissue of a buried plant or organism with minerals in the groundwater. Replacement frequently results in the generation of an exact replica of the original hard part. One absolutely mind-boggling example of replacement is the petrified wood exposed in Petrified Forest National Park in northeast Arizona. Another is at Theodore Roosevelt National Park near the western edge of North Dakota. Here's an example of petrified wood. Slow replacement of wood tissue with silica in groundwater enabled the pigments in the wood to stain the silica crystals as they crystallized. The result is the petrified wood you see preserves the appearance of tree rings, natural coloration, and in some cases, even bark, despite there being no organic tissue preserved. Everything that's there is instead secondary silica that replaced the original material. The third way organic tissue can be turned to stone is carbonization. Soft tissues of both plants and animals under the right anoxic conditions, upon burial will lose their volatile components, which are oxygen, nitrogen, and hydrogen, during decay. Here's a great example of carbonized plant. What's left behind after this process of outgassing is carbon in a relatively stable form. Although frequently compressed to paper-thin thickness, the soft parts are preserved. These are leaves that are 300 million years old. The classic example of preservation by carbonization are the fossils of the Green River Formation of Wyoming. These were deposited over a 5 million year interval beginning 53 million years ago during the Eocene. The carbon films are just amazing. To create these fossils, there are two principal fossil layers. There had to be a chemically complex environment in the lake that the Green River Formation was deposited in. So it does take a unique environment. But when those environments occur, they produce spectacular fossils. To some degree, carbonization is part of the process of converting peat to coal. Of all the practical geology components of fossils and fossilization, perhaps the most practical is the creation of fossil fuels. Fossil fuels like coal and petroleum are the alteration product of buried organics, fossils, but with a very altered chemistry and appearance. Another group of fossils are trace fossils, which provide evidence about past life. In most trace fossils, none of the original organism or plant is preserved. But that doesn't mean the trace fossils are any less important in understanding past life. The first group of trace fossils are molds, casts, and impressions. Many times, the hard part of an organism, for example a shell, will dissolve over time. If the length of time it takes to dissolve the shell is enough to allow the surrounding material to get hard, the void space or hole is left where the shell dissolved. This is called a mold. There are two types of molds. One that forms around the outside of a shell. Here is an example. The other is an internal mold that forms on the inside of a shell. If the mold, that is the void space created when the shell dissolves, is filled with sediment and that sediment hardens, this is called a cast. In some cases, a cast can be an excellent representation of the original organism. Here are two internal casts of bivalves. On this second one, you can see the growth lines of the animal shell. Yet both of these are entirely made of mud. This is a concept that's important in a dentist's office. A dentist will put plaster or plastic substance around a tooth to make a mold or impression of that tooth. The dentist can then fill that mold with other material to form a cast, which can be used to cap the tooth or replace it. So the concept of a mold and a cast is a familiar one. Artists also use the mold and cast concept to make bronze busts, first constructing a sculpture of something that they can easily work with, like clay. Then they make a mold by putting the sculpture in plaster. When the plaster hardens into a mold, they pour in the bronze and create a cast, which becomes the bronze statue. Impressions are a more subtle feature. These are indentations formed when leaves, for example, are gently pressed into mud. A mold is a fairly deep imprint, while an impression is just the slightest depression of the bedding surface. There's not enough displacement volume to make a cast. It's amazing how these very fragile forms can be preserved in the geologic record. A second group of trace fossils capture movement, whether in the form of tracks or trails. Tracks are discrete impressions made by walking, such as footprints in mud or snow. Tracks can be made by any animal that walks. This is the track of an Ankylosaurus from the Jurassic of Massachusetts. Can you see that it's actually a cast of the footprint? It's a positive feature, not an imprint. Trails are a continuous impression. They're made by an animal that crawls. Snails leave behind trails in soft sediment of a lagoon. Some animals will leave behind both tracks and trails. For example, an alligator will leave behind individual footprint tracks and the trail made by its tail. There are also three-dimensional tracks in the form of burrows, which are holes or tubes excavated in soft sediment. Worms leave behind burrows. Frequently, burrows, such as worm burrows, are infilled with sediment and preserved in the geologic record. They can provide an important record of paleoenvironments. There are also some large animals that leave behind very impressive burrows. A series of burrows in the Badlands called the Devil's Corkscrew were excavated by late Oligocene-age beavers from about 25 million years ago, called Paleocastor. A last type of trace fossil are the coprolites. This is fossilized dung. They are classified as a trace fossil because they don't include any body parts from the organism. What they do provide is evidence of paleo diet and paleo environments. Just for the record, coprolites don't smell and they don't have germs. Usually what happens is the coprolites are buried and replacement occurs, so they are silica, not organics. But it's weird that of all the things that are preserved from deep time, it's dung that's the occasional winner? Or maybe that's just another strange lesson from life in general. Paleontology is really an amazing, inspiring, and ever-changing field of geology. The next time you find a fossil or see one in a museum, think about not just the organism that's been preserved. You can also think about where it lived, the environment and conditions that existed at the time, and of course, what were the processes that resulted in its fossilization. There's a lot of geology in each fossil for you to think about. Paleontologists are fossil hunters, where the high point is finding a specimen that's as rare and unusual as possible. In our next lecture, we'll talk about how you can find fossils.